Will the subject maintain laboratory and fieldwork safety? For assessment number 1, what you'll need to do is to complete a risk assessment. So if you look at the question in regards to risk assessment, this is question 14 and question 15. So in question 14, we're assuming that you're working as a team. So normally these workplace safety teams are called a designated work group. So it'll be a team of maybe 3-4 people working in a particular area, and they'll meet together to discuss safety issues. You'll also have within the laboratory somebody allocated as the health and safety representative, so that, if you like, that'll be the team leader in this case. Now, the hazard that we'll be considering is hazard associated with working with E. coli 0157, and we're going to look at a particular case study. So we're going to assume that we're working in this laboratory described in case study 3. So we'll look at that case study now. So if you go to the resources page, you'll find a document called 4 Laboratories Associated Cases of Infection with Escherichia coli 0157. Now 0157 is a particularly nasty type of E. coli. It causes a fairly severe infection, not just a normal food poisoning. It'll invade through your intestines into your bloodstream and release a toxin that can cause internal organ damage, and it can also result in death. So what we're going to do is, assuming that we're working in this laboratory number 3, so what's happening in laboratory number 3 is one of the lab workers has already become infected with E. coli 0157. We can tell that because of the symptoms. So we've got typical blood in the stools, which indicates that internal bleeding as a result of infection with E. coli 0157. Now, that lab worker didn't wear gloves, but she did wear a laboratory coat. She was working with E. coli 0157. There wasn't any mixing or vortexing involved, and there were other people in the lab also working with the E. coli 0157. They did wear gloves, but they're also wearing gloves when using computers and when using taps. So possibly they've spread this E. coli 0157 to the computer and also to the laboratory environment, such as the sink and handles. Okay, they didn't routinely wash their hands, so again it means that they may have spread some of that E. coli throughout the lab. It also talks here about the person ate hamburger. Well, they're only talking about that because in the past the E. coli 0157 has been associated with raw meat. So just discounting that as a possibility, so we know that the E. coli infection happened while the person was actually working in that laboratory. Okay, so what we need to do next is now fill in the risk assessment form. So the risk assessment form is called Classroom Task Risk Assessment Checklist. So we can start to fill in some details. So in terms of the activity in the case study, we can see that they're working with E. coli 0157. The analysis will be conducted as a team, so we'll just say it's the designated work group, and we're just going to use today's date. So it's 20th of the 10th, and then we need to work out what sort of hazard are we dealing with. Is it biological, chemical, environmental? So it's quite clearly it's E. coli, so it's going to be biological. So some sort of disease, some sort of virus. So in other details we can actually specify that we're working with E. coli 0157. Okay, now scrolling down, the next part we need to fill in is we need to do an assessment of the risk. To do the assessment of the risk, we use the table below, and we're going to do this twice. We're going to do it before we implement any controlled measures, and also another one after we implement control measures. So the first thing you need to think about is the consequence. What's the consequence of catching or being infected with E. coli 0157? So I've already mentioned it can cause internal bleeding, it can cause damage to your internal organs, and has in some cases resulted in death. So we're going to say that the consequence is quite major. You can have major damage to internal organs and possibly even death. Now, next thing we need to think about is the likelihood of catching E. coli in that laboratory described in case study 3. Now, because they're not wearing gloves, and because they've allowed contamination of the computer, and possibly allowed contamination of the sink and taps, it might be quite spread in that laboratory. So we're going to say it's going to be quite likely that somebody walking into that lab, if they're following the current practices, there's a good chance that they will also be infected with E. coli 157. So if it's quite likely and the consequence quite major, that means now what we're dealing with is a very high risk situation. So if you've got a high risk situation, it means that you need to take immediate action to control the hazard, and you need to start implementing the hierarchy of controls. So if we look at the next step, step 3 is controlling the hazard, and what we're going to do is go through each of the suggestions in the hierarchy of control and see which control measures we can actually use. So here's our hierarchy of control. So ideally what you would like to do is eliminate or substitute the hazard with something else. So for example, if we were staining some specimens in the laboratory and I was using a stain, say it's a red stain, and it's been found that this particular red stain is potentially carcinogenic, and there's an alternative red stain which is not carcinogenic, well, one thing I can do is just eliminate and substitute that red stain for the non-carcinogenic stain. So sometimes we can eliminate or substitute and get rid of the hazard that way. In this case we cannot, because the lab is designed to be a microbiology lab, so they normally work with E. coli, so we can't eliminate E. coli 157 from the lab. That's what we're actually testing for, that type of bacteria. What we might be able to do is to work safer or maybe isolate. So in terms of isolate, you might be able to use a separate room, so there might be an area allocated just for working with E. coli 157, so you can minimise the spread of E. coli 157 to other areas. Or you might invest in a biological safety cabinet class 2, and that way you can contain the E. coli 157 only within that safety cabinet. So they're all examples of isolation control. The next type of control we can think about is administration. So in terms of administration, you can provide some training or some retraining of the laboratory workers to make sure they're following correct handling techniques for when dealing with microbial samples such as the E. coli. You might need to invest in signage to indicate that there's biological hazards in the lab. You might also have signs reminding people to wash their hands, to wear gloves. You might need to look at your operating procedures, your standard operating procedures, look at the rules that are in place, make sure they're up to date, and update any procedures, any rules as need be. And also in terms of administration, you might need to have additional supervision of the lab workers to make sure that they're following all the requirements. Then the last mechanism of control is using personal protective equipment. So yes, we're going to use those. We're going to make sure everyone's wearing a lab coat, make sure everyone's wearing gloves. So in that case 3, not everyone was wearing gloves, and make sure everyone's wearing the safety glasses as well. Once we've completed that, so we've made up a number of suggestions as to how we can actually control the hazard. So imagine we've got all of these control measures in place. So after we've got all those control measures in place, what we then need to do is then complete the risk assessment again. So again, we can think about the consequence. Well, we're still dealing with E. coli 157, so the hazard hasn't changed, so it's still a major problem. If you get an infection with E. coli 157, it's still a major problem, so that doesn't change. What would change is now we haven't got people not wearing gloves, we haven't got people not washing their hands, we've got new procedures, we've got supervision, we've got training. So the likelihood of people then catching E. coli 157 should be greatly reduced, and hopefully it's very unlikely or pretty rare. So we'll go with rare, so that would make the hazard risk now classified as low. So we've gone from a high risk situation to a low risk situation.